All right, good evening, everybody, and it's great to see you again. We're here with our 7 of 7 of our virtual seminar series webinar presentations. So thank you for joining. My name is Marianne Whitman. I'm the Executive Director of the UC Santa Barbara Natural Reserve System. And when they say save the best for last, well, that's definitely true for tonight. Tonight we're being hosted by the K.S.N. Rancho Marino Reserve. And we are so lucky to have Christy Bell of UC Santa Cruz presenting tonight. And I just can't wait to see her talk. First, I just always like to introduce the UC Natural Reserve System a little bit. I think a lot of you who are out there tonight have been joining us for the whole season. But in case anyone's new, what is the UC Natural Reserve System? Well, it's a very special thing. It's a set of 41 sites distributed across the state of California. And all of these lands are protected explicitly for the purpose of research, university level education, and public service. Altogether, there's over 850,000 acres that comprise the UC Natural Reserve System. And we here at UCSB, we manage 7 of these 41. And so if you've been joining us for the season, you've gotten a little virtual tour of all 7 of our reserves, or 6 so far, and tonight will be the last with Rancho Marino. So again, I can't wait for this evening, and thank you again for joining us. Before we get started, I wanted to just talk about a little bit of what the format for tonight is, and what the with how we will do the Q&A session at the end, which is how you, our audience, will be able to get to interact with our 2 speakers tonight. So first, our reserve director from Rancho Marino, Keith Seydel, will be presenting for about 15 minutes or so. And what he'll do is tell us all a little bit about what's going on at Rancho these days. And then Keith will introduce our speaker, Christy Bell. Christy will then speak for about 30 minutes or so on all of her exciting research, which occurs at Rancho Marino and also a lot of other coastal sites in California. And after Christy's finished speaking, then we'll have about 10 minutes for a Q&A. And I will moderate that Q&A session. And this is your chance, the audience, to ask any questions you would like to our speakers. And the way that you can do that is by using the Q&A button that's on the bottom right-hand corner of your Zoom window. I think we're all familiar with those Zoom windows now. So please do submit your questions. We can't wait to hear from you. Last thing is that this webinar tonight is being recorded and will be available on the UC Santa Barbara Natural Reserve System YouTube page tomorrow. So if you didn't catch all of it, or if you want to share this with anybody, then that link will be sent to you in a follow-up email. And so with that, I'm going to turn it over to you, Keith, and why don't you take it away from here?
All right, sounds good. Go through?
Looks good. You're a little quiet, Keith.
Did that go through?
Yep, looks good. Thank you.
Okay. All right. Good evening. I'm Seydel. I'm the Reserve Director here at Rancho Marino Reserve, the Kenneth Norris Rancho Marino Reserve. And I wanted to welcome you to the final seminar of the 2001 seminar series. And I'd also like to welcome you to the Rancho Marino Reserve. I'm going to give you a quick tour of the reserve and let you know a little bit about what's going on before we start the seminar. Rancho Marino Reserve is named after Kenneth Norris, who was one of the founders of the UC Natural Reserve System. Back in the 60s, he realized the need for protecting natural habitats for university use. He saw that the open land near his research areas were quickly becoming housing developments, and so he put forth the idea of the reserve system. Let me see here, there we go. His conservation ideas helped establish the UC Natural Reserve System in 1965, setting aside land for teaching and research for our future generations. What started with 6 reserves in 65 is now 41 reserves, protecting ecosystems from Mount Lassen in Northern California all the way down to the deserts of Southern California. Rancho Marino Reserve became part of the NRS system in 2001 and encompasses 600 acres. The reserve is located on the Central Coast, just south of the city of Cambria. In this historic photo, it's kind of taken from the north looking south, and you can see the area of the reserve there outlined in red. For reference, the blue line is Highway 1 running south just on the other side of the ridge, heading south there towards San Luis Obispo. This next photo, this is actually taken recently, and so you can see the growth of Cambria there on the edge of the reserve, and actually the growth of the pines as well, if you look carefully there. This shows you, this is present day. This was taken recently of what the reserve looks like. And one more here, this is a fisheye view from offshore looking onshore, and this allows you to see the full coastline of the reserve from Cambria up in the north, down south, heading towards Harmony Headlands down there. So one of the unique things about Rancho Marino is it actually encompasses 4 different habitat zones as you move from the ocean to the top of the 700-foot ridge. This allows a wide variety of research to be done at our location and attracts researchers and classes from around the nation. About half of our current research here at Rancho Marino is marine-based. The 2 miles of coastline provide researchers with an undisturbed rocky intertidal habitat for monitoring and experimental studies. In addition to this protected intertidal, the ranch is fully within the White Rock State Marine Protected Area. And so that allows subtidal researchers access to the state protected water that range from the coastline all the way out to a bit over a half a mile offshore. Along the bluff tops, as you move from the intertidal, you're into the California coastal prairie. And this takes up about 225 acres of the reserve. This habitat has been classified as critically endangered since it's disappearing quickly. It is the most species-rich grassland in North America, and unfortunately, it's also the most urbanized vegetation type in the US. Historically, this land on Rancho Marino, some of it we use for row crop, and there was a dairy operation here from the mid-1800s until 1940s. At that time, the ranch was transitioned into a cattle operation, and the grazing is actually ongoing now. We have long-term experiments on the coastal prairie, studying the effect of cattle grazing on native versus invasive plant interactions. And the grazing is also used as a fire abatement tool, since the cattle do a great job on keeping those grasslands low. As we move up the foothills from the ocean, the habitat turns into coastal scrub. Excuse me. There are several different types of sage growing in this area on these steep slopes. This area encompasses about 40 acres of the reserve, and it's pretty isolated to the steep slopes, mostly along the southern border of the reserve. The 4th habitat, and one of the ones that we're really known for, is our coastal forest. This is made up of Monterey pine and coastal live oak. The reserve has about 220 acres of this mixed forest, mostly in the northern part of the reserve, and from 100-foot elevation up to the ridge at 700 feet. Although Monterey pine is one of the most cultivated trees around the world for lumber and pulp, the wood you get from Home Depot, that 2x4 you go to, is probably Monterey pine. There are really only 3 native stands of Monterey pine left in the US. The Cambria forest is the southernmost stand, and Rancho Marino protects about 10% of that Cambrian forest. This protection allows for research on the ecology of this rare native stands of pines. Now, our forest also, it's mixed in with live oak. So it's not just a pure forest of Monterey pines, it's a mixed forest. And if you joined us last year for our seminar series, you'll remember that as conditions become warmer and drier, the oaks start to outcompete the pines. This is evident as you hike up to the ridge of Rancho Marino, going from the cooler, fog-influenced area at the lower end elevations to that hotter ridge line at the top. I missed all that. And there's still a little, not much known about the basic demography of the Monterey pines. So we're always encouraging more forestry work on our pines. We're just now trying to get the Cal Poly Forestry Department in here to actually do some work here as well. The reserve also is home to 2 stock ponds and 2 seasonal streams that gives just a little bit of a flavor of freshwater habitat to the reserve. See, the classes that visit the reserve are mostly from the UC system, but also from California state colleges and the California community colleges. Most of these classes are unfortunately only here for a couple of days, mostly on the weekends, but there are a few of them that manage to spend a week or more here. Approximately 3/4 of the use here at Rancho Marino is for the classes, and the rest is for university researchers. Our researchers are mostly from those same institutions, but there's also individuals from colleges around the nation. In the last year, the research at Rancho Marino has included research on the intertidal and the subtidal, research on the frogs in our ponds, the beetles in the grasslands, and insect surveys throughout the auroras. We've also had research looking at the scrub plant communities on the hillsides, as well as the pine research in the pine forest. The facilities here at Rancho Marino are anchored by a double-wide modular home and a classroom area that can seat 30. We have an outdoor seating area for 50 and a large camping area that can house 40. Although we don't have a Yelp review, we are rather proud that one of the researchers last year who works at many of the UC reserves voted us the best place for your morning cup of coffee. As a researcher myself, something that I've always been interested in has been long-term data. Previous to being the reserve director here, I worked with the long-term ecological research group at the University of California, Santa Barbara. The natural reserve system is a perfect setting for long-term studies because the land is protected, and thanks to the insight of people like Kenneth Norris, this gives researchers the peace of mind that their research sites will be here as they left them, even if they come back only once a year. We are very fortunate to have several ongoing long-term studies here at Rancho Marino. The original reserve director here, Don Canestro, started a Monterey pine study in 2002. It's ongoing to this day still. There were originally 4 transects within the forest, covering 8,000 square meters of habitat. Every tree in that area was tagged and has been followed. Every new recruit in that area has been watched and measured as it grows, and every death of each tree in there has been recorded as well. This was a really large project, and then actually with the help of a Cal Poly researcher, who's now actually at the University of Nevada, Reno, it was expanded to even more plots covering even more of the forest. This is now an amazing data set covering nearly 20 years of the life cycle of the pines in this area of the forest. And since Monterey pines typically only live for 50 to 80 years. So we're getting up there. So hopefully by the time I retire, we'll have close to the full life cycle of pines data. Another long-term data set that was started here at Rancho Marino is an experiment looking at the grazing pressure on the grasslands. This experiment was started by Carla D'Antonio at UCSB in 2005 and included grazing exclosures along the coastal prairie. Although the regular data for this experiment is no longer being taken, the exclosures remain in place, and that maintains 15 years of habitat of grazed versus ungrazed grasses. And just last year, one of our graduate students was rather excited that this was available for them to utilize and have include that as part of a replicate for their research. So one of the things that I've done, I've been here at Rancho Marino for 2 years. One of the things I've started is photo monitoring to try to get some long-term data for the reserve. We now have a set of trail cameras throughout the reserve to assess the local animal population. And I'm also regularly taking photos at specific locations to watch the seasonal and yearly changes. I'd also really love to add some drone surveys to the forest as well. And we're also updating all of our species lists as we can to try to get that clean and nice so that the incoming researchers know what's available when they come, what's actually found here, and how they can add to those lists. So tonight, we'll hear about some of the other long-term data being collected on the reserve and also along the West Coast from our seminar speaker, Christy Bell. Christy is a research specialist in the Ramundi Lab at UC Santa Cruz and has been conducting research on our coastal intertidal habitat for the past 20 years. In fact, one of the groups that she works with was the very first research group to visit Rancho Marino when the reserve was opened. Christy helped Dr. John Pierce develop a citizen science program called the Long-Term Monitoring Program and Experimental Training for Students, or LIMPETS for short. This was a program developed for the National Marine Sanctuaries. She's also led field teams along the coast to assess the impacts of oil spills, boat wrecks, dredging activities, and landslides. Christy is currently involved in several projects, including long-term monitoring and coastal biodiversity surveys along the West Coast. She's evaluating our California marine protected areas. She's tracking the marine disease outbreaks and black abalone habitat and population assessments. Like many field scientists, she credits a field quarter trip during her undergraduate studies as a life-changing and directing experience. With that, I will let our speaker take over here.
Good evening. Thank you so much, Keith and Marianne, for inviting me to the seminar series. Thank you for the introduction. And it was really great, Keith, to see your presentation tonight as well. I am just a small part of this monitoring program that I'll be presenting tonight and sharing why our long-term data are so important. So back in 1969, there was a massive oil spill on the coast of Santa Barbara, and this really highlighted the need. It was sort of a wake-up call for the need for long-term monitoring data. This also was sort of a pivotal point for the environmental movement and was the seed for our long-term monitoring project. As part of the multi-agency Rocky Intertidal network, we have over 45 partners. We all use standardized methods in order to collect long-term spatial and temporal data. This program has been ongoing since about the mid-1980s out on the Channel Islands, and officially since 1987. There are now more than 250 sites from Alaska all the way down to Mexico, as well as some sites on the East Coast in New Hampshire and Maine. Some of these sites have been monitored for 30 to 40 years, and it's now one of the largest monitoring programs of its kind. In 2012, we were recognized by the Department of Interior with the Partners in Conservation Award. So out of the 41 UC reserves, we are currently conducting long-term monitoring at 6 of them. Bodega, Año Nuevo, this is courtesy of Patrick Robinson, the director there. Rancho Marino, which has become a home away from home for me over the years. Cola Point, Santa Cruz Island Reserve, and Scripps. In addition to these sites that have field support, we are also staying recently at the new Point Reyes field station. We've been staying at Big Creek since the beginning of our monitoring, and as well as Sedgwick Reserve more recently during the COVID pandemic. The reserves provide amazing assets for our program. There's limited public access, on-site protection from poaching and other activities. Their low cost accommodations allow us, helps with the feasibility of our monitoring program. And then now with all these electronic devices that we're using for data collection, it's important to have internet access and electricity. We sometimes use vehicles at Santa Cruz Island and occasionally Rancho Marino and Big Creek. Sites provide help with access and logistics and sometimes field assistance. And I really want to emphasize how important the reserves were to our long-term monitoring program during the COVID pandemic. It really allowed us to continue to work safely and to collect data and minimize data gaps. This wasn't just for our group. Many of the collaborators that we work with were not allowed to travel during the early days of the pandemic. And so we were able to collect additional data for them. So the reserves really made it possible for our monitoring program to continue where it may not have otherwise. So we have the general goals of our marine program are to develop a long-term spatially extensive, feasible funded program that collects baseline intertidal data, to track natural changes within and between communities over a large spatial scale, and to assess impacts, both natural and anthropogenic, such as oil spills, landslides, El Nino events, harvesting, and disease, and to use our data to inform policy and management decisions. So the backbone of our program are really these two complementary surveys. There's lots of other stuff that we do, but I'm going to focus on these two components tonight. So the first one is community structure or core surveys. These surveys target specific algal and sessile invertebrate species that represent different zones of the intertidal. The second type is biodiversity surveys, which is a site-wide assessment of our biodiversity. And this is a photo of Rancho Marino Reserve. We surveyed biodiversity site there in 2021, just this last summer. So before I get into the details of the program, you know, I'm not sure what everyone's background is, so I want to be sure everyone's understanding what the intertidal zone is and why we study it. So the intertidal is the land-ocean interface, the shoreline that's covered at high tide and exposed during low tide. Rocky shores are also the most accessible marine habitat, which fosters a strong public appreciation of these communities, but also makes them vulnerable. These systems are dynamic and biologically diverse communities that is sensitive and subject to dramatic changes from both natural and anthropogenic impacts. And natural variation can be quite high in the intertidal and can occur over the scale of months, seasons, annually, or over decades. So long-term monitoring program is really essential for separating natural change from human-induced change. So just briefly to show you what zonation is, to help kind of explain where I'm going with this. The different levels of the shore are dominated by different species, and the region of the shore occupied by species is referred to as its vertical distribution or zone. So this photograph just seemed to illustrate that pretty nicely. Down here is the low zone. This is dominated by kelps and algae. This is the mid zone, dominated by the sea mussel, Middleus californianus. This is the high zone, here dominated by the barnacles and some algal species. And then this is what we call the splash zone. So this is an area that is primarily exposed to air. Occasionally large waves will crash over it or splash up into it. But this is, for the most part, not a lot of biology occurs up here. The lower zone of the mussel bed is set more by biotic factors such as competition for space and predation. And here you can see the sea star piesaster, which preys on mussels, and that kind of maintains the lower limit of the mussel bed. The upper edge of a species zone is more determined by abiotic factors or physical factors such as desiccation and how long it can tolerate being out of the water. So now that we have a little bit of background on that, I want to talk to you about some of the species that we target in our community structure surveys. So this is just a handful of the species that we survey. There's a complete list of them on our website, which I'll show you at the end of my talk. Again, the acorn barnacles are a species that we target in the high zone. Sea mussels, we target these in the mid zone. And then there's a couple of algal species that I'll show tonight. I'm going to show an example from our northern rockweed, which is a fucus, which these two species are more in the mid zone, and these are kind of lower high zone to upper mid zone species. So we monitor those types of species in these permanent plots. These photo plots are 50 centimeters by 75 centimeters. They're marked by stainless steel bolts. And there are 5 replicate plots for each of the species that we monitor at a site. So for example, there would be 5 barnacle plots, 5 mussel plots, and 5 of the rockweed plots. There could be more species at a site, but just to give you an idea of how that looks. Then we use these grids that have 100 points, and we lay these on the plot, and then we count everything at the intersection points. And then that data gets entered directly into an iPad. And we can track how these plots and communities change over time. So here's an example of one of those plots, just to visually see it changing over a 6-year period. So this is one of the northern rockweed plots. It's just one plot. It's the same one in each picture. This is what it looked like in 2005 when the rockweed cover was really high. The rockweed declined, another alga came in. It got covered in sand in 2007. That scoured out the plot. Then this ephemeral species that comes in after disturbances, the sea lettuce ulva came in. And then we start seeing some more of the fucus returning to the plot. So there can be dramatic changes over short periods of time. Okay, so now I wanted to show you a way we can look at large scale temporal and spatial data for our sea mussel plots. So on the y-axis is site, and this is from Washington in the north all the way down to Southern California. And these are all of the sites along the coast where we sample mussel beds. On the x-axis is year. And so you can see that kind of varies, like depending on when the site was set up. So sites that have been sampled longer, you'll see data all the way across. And again, the program, remember, started in Southern California. And then the color scale, so there's 0 mussels when it's yellow, and the darker the plot becomes, these darker colors are between 75 and 100% mussel cover. So just looking at this, it's a really neat way, someone that doesn't really look at this very often can straight away see that there's higher mussel cover in the north. Many of these sites have a lot of mussels. And then in the south, a lot of these sites, the mussels have declined and the cover is much lower. Just to highlight one of the reserves, I blew this up so you could see easier. This is Bodega Marine Reserve. The plots, when they're established, were high cover. It's pretty stable. And then there was a marine, it got hot outside during a daytime low tide, which caused mussels to die. So when we sampled, we saw gaping mussels. Once the mussels die, they're more easily ripped out during winter storms. So we saw a little dip, but you can see the colors starting to get darker again, and the mussel plots are recovering. So another thing we do is we take these panoramas. These are taken from fixed points all across a monitoring site. There's usually around a dozen or so at any given site. And this is a way to document what the site is looking like outside of our photo plots, to kind of put that in context. So if there was a big disturbance after the fact, we could see, oh, this is what it looked like before this event, and this is what it looked like after. So this is our Sea Ranch site. This is what it looked, we sample it every June. And this was June 2015. And then this is June 2016. So this is a site that is really influenced by sediment. There's We often come, that was also the picture I showed earlier of the fucus plot in 2007 that was covered in sand. This is a pretty normal, natural thing that we see. Every few years, the sediment comes in. Sometimes we're there when the sediment's in, and other times we catch it when it's out. So it's good to know these kinds of things about sites. We do other surveys in our long-term monitoring project. We look at barnacle recruitment, like the little larval babies coming in. We see how they change over time. We do mussel bed depths and sizes, and we also collect genetic samples, often in collaboration with researchers at other institutions. Because we're out at so many sites along the coast, it's easy for us to generally add that to our workload. In addition, we do temperature monitoring. So we have these little loggers that we bolt down to the rock. You can see this is like temperature on the y-axis and month along the x. And so when you look at lots of sites, each of these different colors is a different site. There's a bit of variation over that scale, but then when you look at it on an annual basis, they track each other pretty well, even across regions. And so I also wanted to show Bodega Marine Reserve, shown in red, and the blue line is Rancho Marino. And this is the temperature anomaly over these years from 2005 to 2020. And starting around mid-2014, we saw that sea surface temperatures were 2 to 3 degrees above normal. So this is another important part of the data that we collect. There's some special interest species that we monitor as well, and I'll show you some data and give you a bit more information about these in just a second, but we sample owl limpets, sea palms, which are an annual kelp, and then surf grass, ochre, sea stars, black abalone, and red abalone as well. Here's one of our owl limpet plots. This is a close-up showing a large female owl limpet and her territory. And she grazes the rocks with a specialized tooth called a radula, and she scrapes off diatom films and algal films and maintains this territory. And she will actually push out other intruders to maintain that. So we monitor these plots with these, most of them are 1 meter diameter circular plots, and there's 5 at each of our monitoring sites. Because it's the larger females with more reproductive output, it's really important to keep track of these guys that are harvested, and it's usually the large guys that are taken. So on this graph below here, we have the mean number per plot. So there's 5 plots, and when this was established, there were about 40, so roughly 200 at the site. And this is kind of what it looked like going on. And then after this recent marine heat wave and El Nino event, there was a huge recruitment pulse and the population increased. And we saw this at lots of our sites along Central California, as well as some of our southern sites, but not to the same level. Just to look at these data now as far as size go. So here on the y-axis, we have season. This is from spring 2002 when the plots were established, all the way down to fall of 2020. And you can see the size of the bubble is related to the number of individuals in that size class. So the tiny bubble, there's like 1 or none, and then all the way up to about 100. And this is size class along the x-axis. So you can see periodically there were these recruitment pulses where some babies came in and they grew up, survived to adulthood, and contribute to some population growth. And then you're losing some big ones and so forth. So this is a nice way to look at the demography and how the population's changing. But you can see this huge recruitment pulse that happened. And these guys are growing up and the population has really exploded here at Rancho Marino. This is another site that we monitor. This is one of the rare kind of oddball sites. Most of them, like I said, are the circular plots, but these are all sampled in these big rectangular plots. And at this site, they're all in the vertical face. And So we know that the decline that we saw, so we have count on the Y and season along the x-axis. And we had this big drop of, we lost 77 individuals one year. And when you look at the size data, you can see right away that all these larger individuals are the ones that disappeared. So we lost 77 of the bigger females that have the highest reproductive output. So this kind of information is important to resource managers. We let the rangers at the site know about that event. We also monitor sea palms. Like I said, these are annual brown algae. They are very seasonally and they're sensitive to temperature. So total count again is on the y-axis and years along the x. And every spring they get kind of ripped out in the winter and then they grow up and they're abundant in the spring and summer. And then that's the seasonal pattern. This is natural. But then the marine heat wave came along and knocked them down. And this is a site on the Central Coast in San Luis Obispo County that we monitor while we're staying at Rancho Marino. And they are gone. We have not seen any of them in the last few years. And then we also monitor surf grass. This, like I said, is one of the few marine flowering plants. It's a perennial. It's sensitive to oiling, sewage, and heat stress. And it's a really important nursery ground for invertebrates and fish. Along these transects, we take point contact data as well as measure the bed thickness so we can get an idea of biomass as well. And this is kind of what one of the plots looks like. It kind of goes along, again, like related probably to the heat stress event. In our permanent irregular plots, we monitor abundance and sizes of several different modal invertebrates, including sea stars, sea urchins, these two different chitons. This is a Katy chiton and this is a gumboot chiton, as well as red and black abalone. Those plots are fixed. So first I'm going to talk about the black abalone data. So here is the historic range of black abalone. It used to be from Crescent City near the California-Oregon border, all the way down to the tip of Baja California. The current range is from Point Arena, California, down to about Bahia Tortugas in Mexico. We monitor over 100 long-term sites throughout their range. Some have been monitored for 20 to 40 years. A fatal wasting disease called Withering Syndrome caused massive declines on the Channel Islands beginning in the mid-1980s and along the mainland through the mid-2000s. We have not seen massive declines due to Withering Syndrome north of San Simeon, which is indicated on the map by this red line here. This is a photo of a withered black abalone. This is when densities were really high out on the Channel Islands pre-Withering Syndrome, and this is post-Withering Syndrome, where the beach is covered in empty abalone shells. Our data are used to inform resource managers, and in 2009, black abalone were listed as critically endangered on the ESA, Endangered Species Act, and the IUCN Red List. So this is one of our long-term monitoring sites on the northern Santa Barbara coastline. This shows total counts on the Y and year on the X. And you can see there were about 500 individuals before this wasting disease affected the population. The population crashed and we've seen no recruitment and no population growth since. There's just a handful of individuals still left at that site. This is one of the sites on the northern Santa Barbara Channel Island. And we see here, we didn't have pre-Withering Syndrome data. The site was established after that, but starting in about 2014, we started getting recruitment and these guys are surviving into adulthood and continuing to contribute to population growth. So that's exciting. And then these are some of our sites along the Central Coast in San Luis Obispo County, where declines were not as severe as they were in Southern California and Mexico, not on the islands, but you can still see that populations were around 200 and then they began declining. The disease, the wasting just kind of marched its way up the coast and thankfully not much further than San Simeon. In our sea star plots, we count and measure the Peisaster acraceus, and then the other ones, we just count them. these are the ones, there's other sea stars that we monitor at a site, but these are the most common sea stars that we see along the Central Coast here. And some of them are quite rare, but all of these species were impacted by the sea star wasting syndrome. These are counted in our plots. These are the other two Pisaster species, Pisaster barbospinus and Pisaster giganteus. Leptosterius, Henricia. This is Picnopodia, sunflower star, which was decimated by the sea star wasting disease. The bat star, the leather star, Dermasterius. This is a photo from Kit Harma, who was able to document sea star wasting disease up in Washington. And this is really dramatic to see that his this 3-day time series, that a sea star went from this to that. It's pretty awful. So here's a picture of healthy stars, diseased, and juveniles. Well, this is our site near the Long Marine Lab in Santa Cruz, and counts are on the y-axis and season along the x. Sorry about this figure, I just realized that shifted, but this is from 1999 to about 2016. And adult Pisaster, this is radius of over or equal to 50 millimeters, and juveniles, which are less than that size class. And you can see that right there is when the wasting disease started impacting the site. We lost pretty much all of the adults in that size class. And then we had this, a lot of juveniles show up. So it's kind of interesting to see that this wasn't the pattern at most of the other sites, and many of those have not stuck around. The sunflower star, Picnopodia, since the wasting disease came through, we have not seen many. We just got recent word that there's some showing up on the North Coast, which is exciting, just a handful. They were critically endangered. Shifting gears quickly here into the biodiversity surveys, we use a permanent transect grid where we conduct point contact surveys. There's a transect that runs along the top of the high zone, and then perpendicular transects that run out towards the ocean. Along these transects, we do point intercept and score the species that are directly under those 100 points along each line. And in some cases, there's layering, like if there's a rockweed over a barnacle, then both of those species would be scored. The data go directly into an iPad app that we worked to develop, and that has error checking built right in. So that also saves a lot of time. Here's a schematic of what that survey looks like. Again, the high zone all the way down to the ocean. Along each line, we also do mobile invertebrate quadrats. These are chosen randomly. So there's one per zone, one in the high, one in the mid, and one in the low. So a total of 33 for any site. She's doing a mobile quadrat here. We also do sea star swaths where we count sea stars on a meter on either side of every transect, as well as abalone. In addition, we use surveyors equipment to create these topographic maps. This is a topographic map of our survey site at Rancho Marino. And then this is an example from Bodega Marine Reserve, where we can then take our point contact data and overlay it onto the topographic map. So you can see the distribution visually of the species. And here we just have a few of them plotted. You can see in blue where the tide pools are. Mussels are shown in black, barnacles up in the high zone in gray. And so that's just another nice way we can look at data. Here are the results from our recent surveys at Rancho Marino. Rancho Marino has only been surveyed 3 times. We try to survey sites every 3 to 5 years, but it's all dependent on funding. And so Rancho Marino was first surveyed in 2001, then in 2005, and more recently 2021, this last summer. We observed 87 species during our point contact surveys, and these are the most common ones that popped up. And you can see that there hasn't been a lot of variation over the last 20 years for the data points that we have. And then this is the mobile data from the quadrats from those recent surveys. And you expect a little bit more variation because these are all mobile critters, but the same ones are showing up in relatively similar abundance. We also use drones, AUVs, to document bigger changes like the debris flow that happened at Big Creek Reserve. There was big fires in Big Sur in 2020, the Dolan fire that affected a large portion of the Big Creek Reserve, as well as a large section of the Big Sur coastline. And that resulted in January, there was a huge atmospheric river that dumped over 15 inches of rain along that area and caused these massive debris flows to flow out through the watersheds. And this is another nearby site, Mill Creek, showing images from February 2021, when a lot of this rocky shore was buried. And then this is a few months later when a lot of that sediment has already been washed off. We also are able to track and document species range extensions. We photograph, take GPSs, and make sure that the people like John and Vicky verify our identifications for things like this that are actual range extensions. Sometimes we're also lucky enough to document some cool things like these anemones spawning at natural bridges in the fall of 2011. Another time we were out and there was diatom foam everywhere. It was like working in a bubble bath. We documented these pelagic crabs coming ashore. And this is a photo of a little sea star with a tiny arm bud growing out of the tip of one of its arms. So our data, just to summarize, are really important for tracking major declines through either overharvesting or El Nino disease, and as well as informing resource managers about species range shifts, invasive species. We have an invasive species site also where we can get people involved in adding their observations, which is important. Our data have been used to help design the MPAs, to create baseline data for the MPAs, and to also evaluate the effectiveness of the MPAs. And also we've looked at sites where there's urban runoff, and then for endangered species listings, as well as natural resource damage assessments following oil spills, such as the one in San Francisco. This is also on our wasting map, seastarwasting.org. This is a way to get citizen sciences involved with our work. While we do have loads of sites, hundreds of them along the coastline, there are still gaps where we don't, and it really was helpful. People were really interested in this disease outbreak. Everybody can identify sea stars. They're a pretty iconic symbol in the Intertidal, and kids get excited about it. So when they start dying off, a lot of people wanted to get involved somehow. So we created this map so people could submit their observations of where they were finding six stars, and it helped us track the spread of the disease and what species were affected. It also, our program allows for lots of opportunities for volunteers and internships through STEM diversity programs, research experience for undergraduates through the National Science Foundation, as well as we have some really interested groups of citizens. This is our group up at Sea Ranch, and they've been involved in monitoring their retired citizens, and they're really excited about our program and have been helping us there since 2013. So that's a great way to engage people and share our program. Our data are also used to provide context for other researchers, for other things people are interested in. It provides context for PhD theses, master's theses, undergraduate projects, and also provides context for restoration projects, as well as models that we have a grad student in our lab now working on a pilot project to model the effects of sea level rise. So there's a lot of different things that can be done with it. This is our GIS interactive thing on our website that you can play around with too, if you're interested. We have other ways of exploring our data. This is another portal to go into either our sea star wasting map or other ways to look at data. We also periodically put out these little kind of trend reports that kind of highlight some of our findings. These are available to download PDFs from our website if you're interested in more information. And then during the pandemic, it was really clear, so many of these groups relied on field classes for their students, and they wanted to have real data that they could have their students look at for projects. So we made this available so that teachers and students could use our data. We get requests all the time for different things. So just to conclude, our continued long-term intertidal monitoring is critical for understanding the effects of natural and anthropogenic disturbances on intertidal populations and communities, for documenting changes and recovery of impacted species. And marine and other long-term monitoring programs are essential for informing policy and management decisions, which aid in the protection and preservation of our coastal ecosystem. And then Marion had asked me just briefly, for the students in the audience to kind of just briefly talk about my path to where I am now. And in a nutshell, it probably started watching nature documentaries such as Jacques Cousteau with my great-grandfather. As a preschooler, I was determined to become a marine biologist and didn't wait to learn to scuba dive. So my family didn't have the resources to pay for such things. So, but it was good because it instilled a really strong work ethic. I started mowing lawns, pulling weeds and such, and eventually saved up enough money babysitting and to get certified at Bob's Dive Shop in the Central Valley when I was 14. And then my dad encouraged me to volunteer there because he thought, if you stick around enough, it might lead to more opportunities. And sure enough, it did. Eventually, I hung around long enough that I was given a job there, and I worked there all the way through high school. My love of the outdoors, I credit to my dad and my grandma. So I just wanted to find a job where I could be outside. And then I did lots of diving through the dive shop, eventually got certified scientific diving. Don Canester was my DSO. And then I was fortunate enough to have a field quarter, as Keith mentioned, in Mexico, where I worked with a couple of students to study the shelter preference of this pygmy octopus. And then when I graduated, I was really lucky. My timing lined up and Pisco was just starting, and I was able to do an internship with Pete Ramundi. And I did intertidal and subtidal stuff. And when the internship was over, he had additional funding for me to continue on part-time with marine. But that also opened the opportunity for me to work with John Pierce, working to help him and his graduate student, Don Murray, develop the Limpets program. So I would encourage any students that are on tonight to try and discover your passion as early as you can. You spend a lot of time working, so do something you love, work hard, take those volunteer and internship opportunities that come your way. Try to get into field classes. And if you're lucky, hopefully that'll lead to your dream job. I always thought I was going to go back to graduate school after the internship or maybe a year or two of working, but my job has really, it's still to this day, I'll be out there and I can't believe I'm getting paid to do what I love so much. So yeah, thank you to that. And I really just briefly want to just say thank you to Don Canestro. He was a wonderful mentor and friend. And he was a real believer, as Keith showed, in long-term monitoring and a great naturalist. So I wanted to dedicate this to him. And thank you to all of our funders, all the organizations. This is obnoxious, but I just wanted, this is a huge program and we couldn't do it without a lot of support. Thank you to all those that provided access and all the countless individuals, the reserve directors, our monitoring team in the Ramundi Lab, our colleagues throughout Marine, and all the volunteers. So now we have time for questions. Thanks so much again for having me. It was a pleasure.
Wonderful. Thank you, Christy. I really enjoyed that presentation. That was so great. I mean, we're so lucky to have people like you who are passionate and who can keep the kind of long-term eyes on our coastal resources. Without you doing what you're doing, we wouldn't understand how the changes are happening. So thank you so much. Christy, if you want to pull down your slides real quick, and then we'll move into the Q&A session here. And just a reminder to everybody to use the Q&A button on the bottom right to submit your questions for Christy and for Keith. And while I'm waiting for some questions to come in, I'm going to just get started. And I wanted to ask Christy, first, can you talk a little bit more about withering syndrome? What is it, or what causes it? Why is it around? And does it, do you notice there's increases with different kinds of environmental conditions, or is there predictors for it? What is withering syndrome?
Yeah, that's a great question. So withering syndrome is caused by a bacterium that infects the abalone, and then it proliferates inside the animal's digestive tract. So the animal will continue to feed, but it essentially will starve to death. It seems to increase during warm water events like El Nino. And that's one of the reasons why we think San Simeon is kind of, well, it's present, it's in populations north, and we occasionally find withered abalone north of San Simeon. It hasn't been as dramatic, and we think that the buffer of the cold water from the upwelling along the coast of Big Sur is what's kind of kept it at bay. At places in the southern Channel Islands where it's starting to recover, those places, there's also a co-infection with a phage that helps mitigate the effects of the bacterium. So that was discovered by Carolyn Friedman's lab in Washington.
So with the black abalone, it was really interesting to see, basically it was sad to see, and also interesting to see that the black abalone populations would basically kind of crash and then, not really recover. But there was one population you had that did start to recruit and did start to recover after time. I imagine that's really hard to study, but do you have a sense as to why black abalone will not recover or not, exhibit recruitment, and then all of a sudden after a long time they will? Is there something special about that location, or did something special happen that year that supported their populations? Why does that happen?
That is something that we're starting to investigate. So we have seen that some places that the density needs to be high enough for there to be successful recruitment. There needs to be a nearby neighbor for successful reproduction. And some of these areas, there's remnant populations, and some of it could have to do with temperature and hydrology out on the islands. But we are starting to see it at some of our mainland sites, and we're excited to see if those juveniles do survive to adulthood and start increasing the populations at other sites as well.
Yeah. Okay, so here's a question from a fellow NRSer out there, Jackie Sones.
Oh, Jackie, hi.
And Jackie says, awesome talk, Christy. And
thank you.
She says, exciting to hear about Picnopodia sightings in Northern California. Can you say about where they were observed?
I only just heard about it from Melissa Douglas in our lab, who got a report. I haven't read the full email, but it was just in the last couple of weeks, and I believe it was on the Mendocino coast. And I think it was someone from Reef Check that may have found it. That's the first sightings that we've heard about in California since the disease. We've heard of them recruiting up in Alaska and Washington, but until this time, it seemed they were virtually extinct in some locations. Thanks, Jackie.
And we have time for one more question. So I'm going to Mario Castellanos has a question of, would you share a little about the types of undergraduate internships that are offered both for field and data science related? Where can we learn more? Can you connect us with that information?
Yes. So my email is Bell, BELL @ucsc.edu. And we have lots of intertidal opportunities in our lab. It's been a little constrained because of the COVID pandemic lately, but it's starting to get a bit better. And so, yeah, or there's ways on our website that you can connect to other people in our group. But I definitely encourage all of you to do as much volunteering internships as you can, because it changed my life. I'm so happy to be here.
Absolutely. And everybody, I put Christy's email address in the chat, the one that she just read off. So if you want to grab it there. And also, if you want to follow up, you can contact Christy directly, or myself or Keith can also connect you with her as well. So sorry,
there's not more time.
Yeah, sorry to say that we're at 8 o'clock now. So we're going to conclude both for the evening and also for the season. And so I just wanted to say, thank you to Keith for that excellent intro to Rancho Marino. I wish I was there right now. Thank you to Christy for just a really amazing talk, and also just for doing what you do. I'm really impressed by your passion and your dedication, and the data that you have is really helping us keep our coasts, keeping us understanding of our coasts. And lastly, I wanted to say thank you to you, the audience, for joining us this whole season and tonight. It's been a real pleasure for us at the UCSB Natural Reserve System. We really look forward to seeing you again next fall for our next group of virtual seminar presentations. So have a great holiday, everybody, and we'll see you next year, if not sooner. Great, thank you. Good night.
Thank you. Bye.